Carolina. I actually work remotely in Colorado, though, or sort of do stuff from there. So I'm going to talk about Penstemon, which will be maybe the second time you've heard about it in this session, but a different clade. So starting with the big picture, what is parallel evolution, and why do we care, and how does that relate to what I'm talking about? So the basic idea here is you can see that I've made a cartoon phylogeny where there are two phenotypes that show up, a red phenotype A and a blue phenotype B, and you can see that there have been sort of three independent evolutionary events leading to the acquisition of this red phenotype across the tree. So going from a blue phenotype ancestor to this red phenotype. And so this is why it's called parallel evolution, because it's two things going from sort of the same state towards a new state that they're converging on in parallel. And why do we care about this? Because I think it can tell us a lot about the mechanisms of evolution across various scales of biological organization. So we can ask this question, and things that I like to think about sort of how do we look at this at different scales of organization. So are we talking about parallelism at the same level, the same trait? So in the sense that we have red flowers, for example, here in Penstemon emerging multiple times in different lineages. If that's the case, is it also happening at the same pathway, for example, the same regulatory pathway or the same metabolic pathway that perhaps makes floral pigments? We can drill down even further. We can say, is it happening in the same protein? Is it happening, for example, in this enzyme DFR that makes different compounds at the end of this pathway? Is it even happening at the same amino acid? And so these are, how you ask this question sort of also determines somewhat what answer you get. Is it parallel if it's happening only at the level of the trait, or what scale does it have to be? And what can we learn about the sort of constraints that are applied to the evolution of these types of fairly complicated phenotypes by looking at repeated incidences in which they have evolved? So turning specifically to Penstemon, this is a clade called Habroanthus, and it's a subset of the fairly large Penstemon genus. And within this clade, there appears to have been 4 independent acquisitions of this hummingbird syndrome, this hummingbird pollinated phenotype. And so comparing the typical kind of bee-pollinated syndrome on the left to the hummingbird syndrome, you see the bee's flowers tend to be a blue-purple color. They tend to be shorter. They have shorter stamens and styles and a smaller amount of nectar. And the hummingbird pollinated flowers are characterized by this bright red coloration. They're long, narrower tubes, longer stamens and styles, and they produce a larger volume of nectar to satisfy hummingbird pollinators. So this is a case in which we can look at this sort of parallelism in a complex trait that has repeatedly evolved over a relatively short amount of time. So Carey did previously generated an F2 mapping population, did a QTL analysis, and was able to find several loci in the genome that are tightly associated with different components of this hummingbird pollinated phenotype. And so this is in one of these sort of bee and hummingbird sister pairs in this Habroanthus clade, Penstemon virgatus and barbatus. And I have put a box around the flower color locus because that's what I'm going to mainly focus on for the rest of the talk. And that in there is actually a gene that you might have heard about if you were in here earlier, F3'5'H, which I'll go into more detail about in a while. But to take a brief step aside, one of the things that we hypothesize could potentially be going on here is that actually there may be a case of adaptive introgression. So on the left, you can see sort of the picture I showed in the opening slide, which is a case of true sort of parallel evolution. You have these independent sort of replicated acquisitions of this hummingbird pollinated phenotype in different lineages. But an alternative is something potentially more like what's happening on the right, some version of this, where this thing evolved once in one lineage and was then ported over into other lineages via hybridization and introgression. And you could then get sort of the introduction of the underlying molecular mechanisms into another lineage, and they're not really independent events in that sense, right? It's something that was basically an innovation that happened once and was then sort of shared amongst lineages. So we've gone to great pains to try to see if this is happening, and took an approach using whole genome resequencing of individuals sampled from across this Habroanthus clade, and generated a species tree using many loci. So we've done this in a couple different ways, using genomic windows, using, for example, coding DNA sequences, and a couple different inference approaches using the multi-species coalescent and astral, and also like a concatenated matrix tree in IQ tree. The big takeaway here is that we see very strong support for the separation of these bee hummingbird sister pairs, where the hummingbird pollinated species fall different places in the tree. So they're not getting lumped together overall. And I've dug into this more using statistics based on sort of the ABBA type statistics. So this is something called F branch, and this matrix heat map, the intensity of the red coloration is corresponding to the F branch statistic, which basically just tells you how strong the signal is for introgression. So each little box is just a pairwise comparison, and this also can account for internal nodes, internal branches on the tree, and relationships there. So I highlighted all of the hummingbird lineage comparisons with these little asterisks. And you can basically see they're pretty much not a lot going on there. So while we have some places on this matrix that have a strong signal of introgression, they're actually between bee and hummingbird pollinated species, and this is something I'm going to talk about more. And there's really no evidence for hummingbird species introgression. And so I have also looked at this at the level of individual gene trees and sliding window ABBA type statistics along the genome, and really we just don't see evidence for it. So basically it seems like it is a case of a real parallel evolution of this phenotype, which I think is super interesting, and actually for the sake of understanding something about the mechanisms is actually probably more interesting than if it was a case of introgression. So I'm going to focus on this clade here. So this is another pair called Penstemon laevis and Penstemon eatoni, and so this is another one of these sort of replicated evolutions of hummingbird pollination syndrome. And I've jumped into this project since I started working with Carrie back in October of last year. But this is an ongoing project that's been going on for several years, and it's collaborative with graduate students and technician Hoyt there. So here's sort of, it's just some shots of the crew out as on a recent field trip. So the really cool thing about this system, it's in this really interesting area down along the Utah-Arizona border in southwestern Utah, northwestern Arizona. And you can kind of see from this topographic map, but I'll show you a picture. This is a sort of an extensive system of canyon lands. They're basically sandy washes separating large sort of uplifted rocky mesas. And if you zoom in on here and go out there and hike, you can find all across some of these sites there are penstemon growing there in this sandy, rocky environment. And there are hybrids occurring between this bee and this hummingbird pollinated species, and these are sister species, again, just to reiterate that. And they're sampling the phenotype space a lot. So there's a very sort of continuous distribution of phenotypic variation, both in floral morphology and floral coloration, that is found in these regions. And you can see that kind of better than my pictures if we look, for example, at this PCA of the floral phenotype. So this is specifically looking at color and the anthocyanin pigments that are expressed in the flowers. And so on the left, we have reflectance spectra that are taken from just measuring the reflectance of the light off of the floral petal. And on the right is then a PCA of breaking down sort of a set of representative variables that represent aspects of the color, such as like the hue, the brightness, the chroma, as well as these pigmentation. You can see they sort of partition along a couple of principal axes. So PC1 seems to capture sort of something about more or less how red the things are and how much red pelargonium pigment they're making. PC2 is more capturing something about the overall total amount of pigment and the total brightness of the flowers. And just to be clear, these are mainly the product of the expression of these anthocyanin pigments, the production of anthocyanins in the flowers, and these are generated by a pathway which we know quite a lot about. So this is a branching pathway, and you can see that depending on sort of which choose your own adventure branch you go down, you'll end up with a differently colored pigment. So either sort of the red pelargonium and the magenta-ish cyanidin or the bluer delphinidin compounds. And it makes sense that we then see these sort of like competing tug-of-war axes, for example, in the PCA, because there are decisions being made at these branch points that tell you what you get to make depending on the expression levels and the enzyme activities of the enzymes in the pathway. And really the main difference between these is the presence of those hydroxyl groups on the far right around that aromatic ring that tunes the absorption properties of the molecules. So we have for several hundred now, and it's a growing data set of these hybrid individuals and quite a few of the parents. We have whole genome resequencing data at like 5 to 10x for most of them, and it allows us to then look at a lot of things about the genomic, what's going on in the genomes. So I have basically, this is using a large set of SNPs from this data, then using a program called admixture to estimate the admixture proportion between the parents, the Eton and Lavis parents, and you can see that they really cover this sort of almost continuous gradient, like a climb from Eton to Lavis across this whole set of 400-ish individuals. And you can kind of see this play out in the geographic space too, if you actually map admixture proportion. So for example, in particular on the left, this long rocky peninsula in a place called Rosie Canyon, you can see this, I'm plotting the Eton ancestry, so darker is more towards the Lavis parent, red, lighter more towards the Eton parent, and you can see that as sort of it goes along this long peninsula, you shift in a somewhat coherent fashion from one ancestry to the other. In contrast, if you look across, there's this sort of bowl area, and the things are somewhat more mixed together there. You can see this a little better if in sort of the population structure, if you look at a genomic PCA of these same SNPs. So the principal component one is sort of separating the Eton versus Lavis parent, and then PC2 is sort of separating the variation within species. And you can see in particular, like this pink square population, which is that peninsula, really samples across kind of this whole continuous gradient from one to the other. This is a really nice system then to use for what's called admixture mapping, and that's basically just treating these as like a natural mapping population. So we have 417 individuals. We can then look at the signal for individual sort of regions, SNPs along the genome that are associated strongly with phenotypic difference. So I'm just going to show you one of those right here, and this is the fraction of the blue delphinidin compound. And you can see that there's this pretty narrow region with a really tall sort of chimney of statistically significant associated points. Right in the middle of that is this gene F3'5'H, which is one of like the principal decision maker for the flux through the pathway that would direct you to or not to delphinidin, depending on the state of that gene. And I think it's really important to also note that I have looked directly at the sequences within this window to see if they support, for example, pairing any two hummingbird species together, and they don't. So there really is no signal that this is not like an independent acquisition of something happening with this gene. And I think that's super interesting. So moving forward, the things that I'm working on now are sort of takes, what we know is that, okay, it seems like it's really parallel. We have this amazing hybrid zone population, and it seems to work pretty well for pinpointing regions of the genome that are associated with the phenotypic variation. So I'm trying to extend these analyses to additional phenotypes, and also to look more at sort of the variability in local ancestry along the genome and how that's associated with the phenotypic variation. So yeah, looks like I'm done, and thanks for listening.